Hello and welcome back to the SIRIUS short course tutorial series. In this video, we are going to go over the different views in SIRIUS to interpret structure annotation results. The first tab of interest is the Structures tab, which shows a ranked list of database structures based on how well they match against the query spectrum's predicted fingerprint. This is expressed as the CSI Finger ID score, for which higher is better and 0 is the theoretical maximum. CSI Finger ID scores should only be used to compare structure candidates belonging to the same feature. For example, the -12 here is much better matched than the -231, but we cannot compare this -12 to a different -12 of a different feature, for example. The top scoring structures row is highlighted in green. This highlighting extends across all results tabs to visually link the structure and its molecular formula annotation. For some features, multiple structures will have the green highlighting. This is the case if a lower ranked structure is so similar to the top ranked one that it is unlikely that they are distinguishable using mass spectrometry. The metric we use to measure this similarity is a maximum common edge subgraph distance of 2 or less. This is for example the case for two structures differing only by moving a side group. In most cases we can also observe that these structures all have very similar score. The sources part of a structure candidates row shows us which databases this structure is contained in, even if we did not select all of them in the computation. If we click on a source label, the corresponding database entry will open in the browser. Next, we are going to take a look at the colored boxes here, which represent substructures of a structure candidate and help us understand which parts of the proposed structure fit well to our experimental query spectra. Every structure candidate in our list here has a known binary molecular fingerprint. This is completely independent of any spectrum and can be trivially computed if we know the structure. Each position in this fingerprint encodes for a specific substructure. Some substructures will be contained in our candidate structure and some will not. Those substructures in the fingerprint that are also part of a structure candidate, we call them contained substructures, are the boxes that you see in this view. Now, every box has a color and a size to relate the contained substructures to the actual measured data. Based on the query MS2 spectrum of a feature, SIRIUS predicts a molecular fingerprint. This molecular fingerprint is not binary, but instead every substructure gets assigned a probability based on how likely SIRIUS thinks this substructure is part of the true structure we are trying to elucidate. Every box now gets colored based on this probability. An easy way to think about it is a green box means that a substructure was predicted to be in the measured feature and is also present in the presented structure candidate, which increases our confidence that this part of the proposed structure is actually correct. Now that we have understood the box's colors, let's talk about the box sizes. When predicting the molecular fingerprint from the spectrum, SIRIUS is better at predicting some substructures over others. We measure the ability to predict a specific substructure by the predictor quality. Boxes are assigned a size based on their predictor quality. The better the predictor, the larger the box. If a predictor is comparatively bad, its color should be interpreted more critically. Simplified, a large green box is a great sign that this part of the structure is correct, while a large pink box is a sign that this part of the structure is likely incorrect. We can click on a box to see which substructure it represents. By using right-click, highlight all matching substructures, we can overlay all predicted substructures at once to see which regions of the molecular structure candidate fit well to the predicted fingerprints and which are likely not correct. If one of our spectral reference libraries contains spectra for a feature, we can see the highest matched cosine to the right. Clicking on the Show More label will bring up a substructure annotated mirror plot. We can also access this view by clicking on the Substructure Annotation tab. In this tab, we can choose a candidate structure from the candidate table to understand how well it can explain peaks from our query spectrum. A peak will have one of three colors: green if a substructure of the candidate structure explains this peak's mass, blue if no substructure explains its mass but a subformula does, and black if neither subformula nor substructure are supported by the peak. It is very important to note here that this substructure matching is not related to how SIRIUS scores structure candidates. It's merely a visualization tool based on combinatorial fragmentation of the candidate structure. Since combinatorial fragmentation can't take rearrangements into account, they will also not appear here as fragment annotations. This does not mean that SIRIUS cannot annotate structures that form rearrangements during fragmentation. Clicking on a green peak will highlight a potential substructure explaining its mass. The spectrum can be navigated either by mouse or using the arrow keys. If we want to inspect these peak annotations not in isolation, but in the context of a spectral library match, we can select an available match from the drop-down menu. Matches can result from identity spectral library search, which means spectra have the same precursor mass, or analog search. Selecting a peak will automatically highlight the corresponding peak in the matched spectrum that was determined during cosine matching. If the current library match we are inspecting is an analog match, some peak matches will represent shared losses instead of shared fragments. The corresponding losses substructure is highlighted in yellow instead. By pressing Enter or right-clicking on a selected peak, we can bring up a context menu to help find reference spectra from the list that explain a specific peak we are interested in. For example, if we want to find a reference spectrum that also contains this peak, we can filter the list of references for spectra that contain this peak as either any match or fragment match only. We can use this view to gain confidence in the annotation of this triphosphate structure. While we don't have a reference spectrum for it, we do have one for a similar compound and can easily see which peaks and losses they share. Now, let's take a look at an example where the structure proposed by structure database search does not look correct. Here, we can see from the highlighting that at least the upper part of the molecule contradicts the predicted fingerprint and is likely incorrect. In cases like these, we can use MS Novelist to generate de novo structures to give us a better lead. The de novo tab shows these generated structures with a de novo source, and we can immediately see that the top generated hit fits better than our best database hit. We can now again check the annotated spectrum for de novo hits as well and compare against the annotated spectrum of our database hit. Please note that MS Novelist is unlikely to produce the 100% correct structure for true unknowns. It should be treated as a tool that generates a lead structure for cases where the database search fails. Most of these validation steps we have looked at require time and manual effort. If we are using SIRIUS for untargeted experiments with thousands of features, validating them in this way is not feasible. SIRIUS comes with a confidence score for each feature, which is between 0 and 1. The higher the score, the more likely it is that the top-ranked structure annotation is correct. This confidence score takes into account multiple metrics such as how many peaks can be explained by substructures, what percentage of the spectrum's intensity is explained by the molecular formula, and more. SIRIUS supports two different confidence modes, exact and approximate. The exact confidence score tells us how likely it is that the exact top-ranked structure is correct, while the approximate confidence score tells us how likely it is that the top-ranked structure or an exceedingly similar one are correct. We again use the MCES distance smaller than 2 as a metric of that. When dealing with large untargeted experiments, it is recommended to sort all features by confidence and then apply a confidence score threshold that dictates which feature annotations we are confident enough in to spend additional downstream analysis on. A rule of thumb here is that a confidence score of 0.64 was often observed to correspond to roughly 10% false discovery rate. In practice, users should set a threshold that leaves them with an amount of features that the downstream analysis pipeline can still handle.